In this video, we'll be talking about gastrulation, which is the first stage during embryonic development. In the previous video, we have introduced ourselves to the concept of embryology, and we have already mentioned that embryology is the study of development of living organism. And we also mentioned that during embryonic development, we have several steps, which include gametogenesis, fertilization, cleavage, gastrulation, and organogenesis. And today we'll be talking about one of the most important steps in the formation of germ layers, referred to as gastrulation. Now, we all know that during developmental stages, like I've mentioned before, we have the gamete formation called the gametogenesis, fertilization, which is the fusion of gametes of the male and female gametes, which leads to the formation of zygotes. Then we have the cleavage, that is a peculiar cell division that occur during embryogenesis. And we mentioned that we have different type of cleavage based on the type of egg that an organism or an animal possess. We also recall that we said we have different types of eggs, like homolecita egg or isolecita egg, centrolecita egg, telolecita egg, and hololecita egg. We have mentioned that before. And we said we have different types of cleavage as well, where we have the holoblastic cleavage and the meroblastic cleavage. Under the holoblastic cleavage, we have the spiral and the radial cleavage. Under the meroblastic cleavage, we also have different types of cleavage under the meroblastic cleavage as well. And we said that depending on the type of egg that an animal possess, we are going to have different type of cleavage. And also different type of egg, it is on the basics of what? On the basis of the amount of yolk that the egg possess. For example, now in isolecital egg, we have equal amount of yolk, which means yolk is evenly distributed throughout the embryo. You understand? And today we will be talking about the gastrulation in Amphioxus, which is a typical isolecital egg. You understand? Now, like I said a cleavage is a peculiar type of cell division that involve the division of the the division of the cell without the separation of itself, which means the number of cell increases, but the size of the cell does not increase. The number of nucleus increases, but the number of nuclear material per nucleus remains the same. All these things we have actually explained it in one of our previous videos that we have done before. So the word gastrula and gastrulation was coined by Ernest Haeckel in the year 1872, and it means the formation of guts. That is what gastrulation means. Now, let us now talk about the characteristic of gastrulation stage. It is the first stage of cellular differentiation, and the embryonic cell begins to follow in specific layer through morphogenetic movement. It should be noted that during gastrulation, there are movement of cells, and this movement of cells is what we refer to as morphogenetic movement. And during this morphogenetic movement, there is formation of germ layers. Therefore, we can say that gastrulation or the germ layer formation is the stage of embryonic development in which a single layer blastula is converted to a trilaminar structure consisting of outer ectodermal layer, the middle mesodermal layer, and the endodermal layer, which means during gastrulation, one layered blastula is converted to a trilaminar structure. And when we say trilaminar structure, it means a structure with three layers, you understand? And these three layers include the outer ectoderm, the middle mesoderm, and the inner endodermal layer. These are the three layers that are going to arise due to what? Due to gastrulation. And these changes occur through series of cell migration from the surface of the blastula into the interior cells, and arising from each of the germ layer, they ultimately give rise to a specific organ or tissue, which means all this layer, which is the ectodermal layer, the mesodermal layer, and the endodermal layer. Remember that they are still cells. They are not tissues yet, and they are not organs. So we are going to see cells of the ectodermal layer, cells of the mesodermal layer, and the cells of the endodermal layer. They are going to give rise to a specific tissue and a specific organ. For example, the ectoderma will differentiate into epidermal layer of the skin and the neural tissue. That is what the ectodermal layer will differentiate to. The mesodermal layer usually give rise to supportive muscular system, skeletal system, gastrointestinal tract, respiratory tract, while the endodermal layer give rise to circulatory and respiratory tracts, or the respiratory system. You understand? So you see, it is now possible to identify the cells in the blastula from which the germ layer arises and from which the organ of the premolar develops. And this particular method of identifying where does these cells arises from. For example, we have seen that there are cells in the ectodermal layer. We now want to know where does it arises from, or what is this cell going to develop to. The method of determining this is what we refer to as fate map. You understand? So we can say that a fate map is a diagrammatic expression that determines the fate of a cell during embryogenesis. That is basically what the fate map is. And we are going to be doing a particular video on that particular aspect of embryology, which is fate map. Now, we have talked about the first step, the second step, the third one, and the fourth one is that cellular division still continues during gastrulation, but cellular divisions lose synchronization between them, which means cell division are not synchronized. For example, during the early stage of cleavage, we have one cell divides into two, two cells divide into four, four cells divide into 16, and 16 cells divide into 32, called the morula. 16 and 32 cell stage, called the morula. 32 can now divide into 64. Now, when we are now having large number of cells, what is going to happen is that synchronization will be lost. Synchronization will be lost. You understand? And during this stage, there are some things that will be formed. You understand? Remember, I told you that it's a special cell division, which means one cell having 46 chromosomes is going to divide into two cells, and each of these two cells is going to be having 46 chromosomes. This one too is going to divide into four cells, each of them having 46 chromosomes each, and so on and so forth. Now, during the end stage of cleavage, because this is cleavage, you understand, a special cell division. During the end stage of the cleavage, there is formation of some things. And these are, there are some cells that will be lining the blastula. We call it blastula, understand? We refer to it as blastulation, you understand? There are some cells that will be lining it. There are some cells that there will now be some cells that are inside of the what? Inside of the this thing itself, you understand? Now, we refer to them as the trophoblast and the embryoblast. So we refer to the one that is outside, we call it trophoblast, because it is also referred to as the outer cell mass, and we refer to the one inside as the embryoblast. Embryoblast, you understand? Embryoblast. You now see the cavity that is formed around this place. Can you see an hollow cavity that is formed around this place? Is what we refer to as the blastocele. All this information are very, very important because we are going to be using this information during the explanation of gastrulation and field use itself. You understand? All these ones are still under cleavage. So there will be formation of the trophoblast, which is the cells that are lining the wall of the blastula, and there will be formation of the embryoblast, which is the cell inside the blastula itself, and there will be formation of a cavity called the blastocele. You understand? We call it the blastocele. Now, remember I told you that synchronization is now lost, which means this 64 is not going to divide into 128. Rather, it can divide into 98, it can divide into 110, because it does not, like the division of 1, does not initiate the division of the others. You see, during this stage, the division of one cells will initiate the division of the other cells. But after there is a large number of cells, synchronization is lost. And that is what they are trying to say here, that cellular division loses synchronization. And the metabolism change within the cell and the oxidation process becomes the dominant factor. The nucleus at this stage has more impact than before in the process of control of development and shows the chromosome of partner genes and the effect. And in this stage, that is where we'll be talking about the molecular basis of gastrulation itself, because the molecular basis actually arises from the concept of the nucleus and the chromosome. We are still going to talk about that. So these are the characteristics of the gastrular stage or the gastrulation stage. So now, like I have mentioned, gastrulation is the formation of germ layers, and gastrulation occurs due to series of rearrangements that convert a single layer to a trilaminar structure called the gastrula. You understand? And this gastrula, it also has its own specular structures. You understand? Its own specular structure. For example, now the peculiar structure of a blastula is the trophoblast, embryoblast, and the blastocele. But the specular structure of a gastrula is the primitive guts or the arc enteron, the ectoderm, the endoderm, the mesoderm, and the notochord. These are the peculiar structure that are found at the end stage of gastrulation. And at this stage, when a particular structure is now having the arc enteron, the arc enteron, the ectoderm, endoderm, mesoderm, and the notochord. We call this particular structure, we call it the what? We call it the gastrula. You understand? So gastrula is the end product of gastrulation. You understand? So let's now talk about gastrulation itself. The blastula consists of numerous cells and the position of which were established during cleavage. They said during gastrulation, these cells are given new position and new neighbors. And these are like this position and new neighbors is due to the what? Due to the rearrangement of cells. Are you getting me now? Now, the cells we form the endodermal and the mesodermal organ are brought inside of the embryo, like the cells that will form the endodermal and the mesodermal structure will be brought inside of the cell, while the cell that will form the ectodermal layer, that is the skin and the nervous system, will be spread all over the surface, and that is what we refer to as what? The ectodermal layer. You understand? Now, in addition, this stage sets the interaction for newly positioned tissues. Like I've mentioned before, these ectoderm, endoderm, and the mesoderm, they are going to give rise to different tissues and organ in the body. You understand? And that is where we'll be going to the concept of stem cells. You understand? Which are precursor cells. Although we won't talk much about the stem cells, I'm just trying to like explain some things to you. So the ectoderm is going to give rise to the skin and these presumptive nervous tissues. The endoderm is going to give rise to supportive system, skeletal system, maxillar system, gastrointestinal tract, amongst others. You understand? And the endoderma is going to give rise to the circulatory and the respiratory system. So this is the system, the cells, the tissues that will give rise to. Remember that during the formation, the organization of life, cells become tissue, tissue becomes organ, and organ becomes system. So now the three layers are formed and organized in their proper location during gastrulation. They will be formed and they will be organized during proper this thing. Now, the fate map, like I have said, I said the fate map is a diagrammatic expression that shows the fates of a particular cell during embryogenesis. Are you getting me now? It shows the fate of a particular cell during embryogenesis. That is basically what a fate map is. Now, if you look at it very well, they said the fate map of Xenopus blastula is shown in this particular diagram that we have here. So this one is for the frog. This one is for the Xenopus. This one is for the Xenopus. And if you look at it very well, they are colored in different way, right? You will see that we have the green, we have the red, and we have the yellow. So this part that is having less yolk, this part that is having less yolk is what we refer to as, or less cells is what we refer to as the animal pole. The one that is having more yolk is what we refer to as the vegetal pole. And if you look at it very well, they have already put it there that the ectoderm is going to form the epidermal layer of the skin. The mesoderm is going to form the muscle, the kidney, bones, and gonads. The endoderm is going to form the digestive system, the lungs, and yes, those are the things that is going to form. And if you look at this very well, this one also shows the one for frog. You understand? I've forgotten that xenopus is actually frog. Don't mind me. Now you will notice very well that these are marked. The cells which this thing is in blue and green. That is the what? That is the skin and the neural tube. The cells which fit become the mesoderm are in red. The cells which fit become the endoderm are in yellow. Now notice that the cells that will become the endoderm are not internal yet, but it will become internal during gastrulation, because if you look at this particular cell that is here that is going to form the endoderm during blastulation, during blastula, this is a blastula of a xenopus, which is a frog. You understand? You will see that it is not internal yet, but during gastrulation they will move into internal. That is basically what a fate map is, because during blastulation we don't know the fate of this particular cell yet. You understand? But during gastrulation we have already determined that these cells that are here will be the one that is going to the endoderm. We have already known that. And during gastrulation, we are going to see how they will actually move into the what? Into the inner layer, or the what? Called the endodermal layer. You understand? So these are showing the derivative of the germ layers, which we have actually talked about before. Let us now talk about the type of cell movement during gastrulation. The first one I'll be talking about is the invagination. And the invagination is the infolding of region of cells. Infolding. We'll be needing this one the most, especially in gastrulation. We have the involution. Involution is the inward movement of an expanding outer layer, so it's spread over the internal surface of the remaining external cells. Involution. Invagination is the infolding of region of cells. Involution is the inward movement. You see, during invagination, there is no movement of cells. There will just be infolding. You understand? It's just like you bending a rod. Let us take it like that. It's just like you bending a rod. Now, you now see involution. Involution is now movement of cells from one point to another, so that they will spread all over the surface of the what? The surface of the blastula that we call the gastrula now. We now have the ingression. Ingression is now the migration, like the movement of an individual cells from the surface of the layer into the interior. Now, look at this. This is a cell, for example. These are cells. I'm just taking for an example. Now, what is now invagination? Invagination is now the inward infolding of this cell. They will infold, then they are going to form something like a cup shape, something like this. You understand? Can you see that cells are not moved, but they are infolded? You understand? What is now involution? Involution is now the movement of cells from one point to another, like they are moving throughout, so that they will be spread throughout the world, throughout the distance. What is now the ingression? Ingression is now that there will be some cells that are outside. You understand? They will now be moved inside of the embryo. You understand? For example, if the endoderm wants to occupy its tip, look at this. This is a very good example. If you look at it very well, this that happens here, look at it, that form a cup-shaped structure. That is what we refer to as invagination. You understand? This is invagination. Now, the movement of cells within this, like to spread across this particular structure, is what we refer to as what? Involution. Now, there will now be cells that are going to occupy some certain regions. Like the cells that are outside, they are going to go inside. There are some cells that will go from outside to inside. That is what we refer to as what? Ingression. You understand? Now we now have delamination. Delamination is the splitting of one cellular sheet into two or more less parallel sheets. And we have the epibola. Epibola is the movement of the epithelial sheet that spread as a unit to enclose the deeper layer of the embryo. This is also seen in gastrulation and amphioxus, and you are going to see that in gastrulation and amphioxus there will be spreading of what? There will be spreading of cells across the this thing. So this is the introduction to gastrulation. Basically, this is the introduction to gastrulation. So it has been amply evident that in all chordates, the position of an organ forming area in the blastular stage differ greatly from the position of the respective organ in the adults. You understand? And the various parts of the embryo must be moved about, displaced from the original position if they are to arrive at their various final position. This displacement of parts takes place in the process that we refer to as gastrulation, and this gastrulation is referred to as formation of germ layers, which involve the movement or the rapid movement of cells. You understand? So this would be firstly described as it occurs in Amphioxus. That's the first one we're going to describe. Then we'll move to the amphibians. From Amphioxus to amphibians. Then from amphibians, we'll talk about the one for reptiles. Then for avian species, that is aves. Then for mammals. So these are the ones that we'll be doing in our next video. So the next video, we'll be talking about the gastrulation in Amphioxus. See you in the next one. Thank you.